To finish this section about somites, we're going to talk about the somite and the epithelial mesenchymal transition. Another component that we haven't talked yet is the epithelial mesenchymal transition, or EMT. And here in this diagram, you see an epithelial, as you have here before in cell biology, anatomy and physiology, or other courses, that the normal characteristics that have junctions in different regions of the cell, and these junctions allow to the epithelia to know where is apical, yeah, the top, where is basal, the bottom. And what is happening is that as they have changes in the gene expression, all these characteristics disappear, and they allow them to break out from the epithelial and go and become mesenchymal cells that are mobile. Yeah, so they lose their orientation, they lose the cell contacts, and they adopt mobile characteristics that happen in mesenchymal cells. So this is very interesting because why? Because one application is that something similar is what could happen during sometimes in cancer, that the epithelial cells can lose they turn into a different type of cells, and they start migrating from the original tissue. In addition to that, we can also have the opposite, the MET, that is mesenchymal epithelial transition, that the mesenchymal epithelial, they change their gene expression, they lose the gene expression of a mesenchymal cell, and they return to the epithelial, and they start developing all these junctions, and they go and attach in the correct places. So two type of transitions: epithelial mesenchymal and mesenchymal epithelial transition. Okay, so something that I didn't tell you before about the somites is that the epithelial mesenchymal transitions are very important here. Why? Because here I have four diagrams. Here in A, a 2-day embryo. Here in B, a 3D embryo. For a 4-day embryo, and here the late of the 4-day embryo. And as you can see, you have the neural tube as usually, the node occurrence usually, and here you have the scleroton progenitors, the somites. And what is going to happen is that while the scleroton is forming here, accompanied by all the developmental process that we talked before, we have this epithelial mesenchymal transition. Yeah, so you see that the cells start losing the contact, start losing the apical and basal polarity, and start migrating to other tissues. And you can see that happen especially in the late day 4. Okay, and this is how myoblasts, yeah, the muscle precursors, are formed. Okay, we finished the somites and the epithelial mesenchymal transition. Now we're going to talk about the kidney development, that is a model for organogenesis and this double induction process. In this section, we will talk about the urogenital system. In particular, we're going to talk about how the kidney formation and how these major organs are derived from the intermediate mesoderm. And we are not going to talk about the gonad development. We're going to mainly focus on the renal system development. And if you remember from high school or from anatomy and physiology, the unit structure is called the nephrons, and here you have collecting ducts that are called ureters. And we're going to show how both produce signals to promote the development of the other one, and what happened during evolution with these things. So the kidney is mostly filled with 1,000,000 nephrons that are the functional unit of the kidneys, and each nephron has more than 10K cells of 12 different types. So we already talked about the paraxial mesoderm. Now we're going to focus on the intermediate mesoderm. We're going to mainly focus on the kidney in this section, and we are not going to talk about the gonad development for this reason. So as we watched in previous videos, You have the epiblast, and in the epiblast you have the primitive streak that is a transient structure that allow you to divide the midline, the anterior, and the posterior in mammal and chick embryos. And from here you can see the different structures of the mesoderm. We already talked about the paraxial. We briefly talked about the lateral, and here you can see the intermediate mesoderm. And from here you see the somites here, and close to there you have the uteric bud and the nephric duct. And here you're going to end up forming the nephrons, yeah, and the renal system. That is what we're going to cover in the following slides. So in the following slides, I'm going to show you the kidney development is divided in three main sections: pronephrons, mesonephron, and metanephrons. And this happened in mammals. And during evolution, there is small changes, but the idea that is that as the pronephrons develop, yeah, it go to apoptosis. Yeah, and then the mesonephron start developing, as you can see here, in a sequential way, it start developing, and this one start developing too. And later on, the first mesonephrons start degenerating also, and you start generating the metanephric one. And at the end, in mammals, you end up degenerating both pronephrons and mesonephron, and you only keep the metanephron that it will develop. Yeah, so okay, so what I show you with this diagram is that there is a sequential development and degeneration of the pronephrons and metanephrons, and how this degeneration occur through apoptosis. Yeah, while there is an induction of the ureteric bud and the metanephric mesenchyme during kidney development in mammals. This happens in mammals. In other organisms, in other vertebrates, it happens slightly different. So in the case of the fish, yeah, and some amphibians, the you only go through the pronephron stage. In other amphibians, that have permanent kidney, they destroy the pronephrons through apoptosis, and they keep the mesonephrons. And in the case of some reptiles, birds, and some mammals, they can keep some of the mesonephron, yeah, or in the case of some mammals, you can completely destroy it and keep this one, the metanephron. Okay, so there is a lot of variation, and it's very interesting to analyze this from an evolutionary perspective, these three different stages. Okay, I know you watched already a video about this question. Does ontogeny recapitulate phylogeny? And you have seen this picture that are, like if you see it, you say, of course, yes. And the problem is that they are diagrams. As you're watching the video, they actually, the development, yeah, this is a historical hypothesis that say that the development recapitulate the successive stages of evolution. Yeah, so what you see during development is what is going to happen. Yeah, so they stop here, so now they continue here, and then they continue here. Yeah, so it's like a building that you're adding more and more floors. However, what we found is that the development has more variability than we expect. So let me show you another diagram in the following. So what Haeckel, that he was a very smart guy, but he got too excited with his ideas, and unfortunately they were not correct. What he was proposing is that for example, this is the embryo and this is the adult. What he thought is that the embryo and the adult of the fish, what you're doing, just when there is a frog, you add a new stage, and here when you have birds, you add a new stage. And what we have seen is that is an example where there is no recapitulation, where the embryo's development is increasingly diverse. So you can sometimes repeat something that happened in some stages, but what you see is that the neural plate can be formed in different ways. It's not fish, frogs, and birds are going to make it in different ways. And I'm just showing you pieces, important pieces to show you processes that are happening, but the ontology doesn't recapitulate the evolution. Okay. Now, very quick, I'm going to talk about in a little bit of detail what happened during the 3 stages. So the first stage, you find it here. Yeah, and in yellow you see the pronephros that are forming, and you see that the pronephic duct start forming, and some cells start migrating toward the tail. Yeah, and start producing this tubule. The next step that happens is that the tubules degenerate. So here again, through apoptosis, they degenerate, but you still keep forming and forming and forming some more, and you start forming here the mesonephrons that you see here, and some of them, these one are connected, these one are not connected yet, and you also produce some of the stem cells that are going to carry the sperm, that are going to become sperm carrying tubes. Okay, so we already degenerate this. We already degenerate, like we're going to start degenerate these ones, and we're going to start focus on the metanephric. Yeah, that are the ones that are going to remain. So we need to expand it, and we're going to focus more on this in the signals that are happening here. That is the metanephron. And here, this process of the metanephron is a process of double induction. We have talked before about one process of induction, yeah, a tissue induction, that you have a group of cells that are inducing a tissue to produce changes, while the other one surrounded is a responding tissue. And if you remember, this is how we found the frogs and the transplantation experiment, that you take one piece and move it into another one, and you end up with a larvae with two heads. And yeah, because we're able to produce two regions where there are induction of the process. Here we have another example of induction. So let me show it to you. So here you can see the metanephric development, and here we will see the mechanism of reciprocal induction. So instead of having only one tissue responding, you will have both inducing and responding to the other one. So there are 5 steps that I'm going to explain in the following slide, and here you have the first 2. I'm going to start telling you that there are 2 main components. As I said to you before, you have the nephric duct here in yellow, and here there is the formation of the metanephrogenic mesenchyme form. And what is this going to do is to secrete a molecule called the GDNF that is going to attach to these red receptors. Yeah, and this is going to produce an induction of the euteric bath. Yeah. So here the nephric duct is going to form this, where the red receptors are going to attach. So now we're in this third step, and as you can see here, the euteric bath attach here. And what it's going to do is, do you remember that there was apoptosis here before? So to prevent the apoptosis, this euteric bath is going to secrete FGF2 and BMT2 to prevent apoptosis from the metanephrogenic mesenchyme. Now, here in the fourth step, and I know if you remember the epithelial mesenchymal transition, and I told you that also there is mesenchymal epithelial transition, we're going to, now this is going to happen, the MET. So the euteric bath is going to secrete another molecule that is going to induce the mesenchyme cells to aggregate and become epithelial. So all the cells that they're well mesenchymal cells, now they're going to become epithelial. And finally, the fifth step. Now, so in the same way that we produce this induction in the frog, here we're going to do this multiple times. So the red receptors are going to appear, there will be induction, they will prevent the cell death, and they will be creating more and more and more branching and creating these units of nephrons. Yeah, and there will be differentiation and growth of the euteric bath, and this is in a very summarized way what happened with the nephrins. Okay, and with this, we finish the kidney development as a model of organogenesis, in particular for reciprocal induction. So I want to give you 5 ideas before you leave. So the first one is that two tissue layers forming gastrulation, the mesoderm and the endoderm, give rise to different organ systems, as we study here, and we talk about some of these structures. The second one is that the mesoderm divides into several tissue structures, and we talk about 4: the intermedia, the paraxial, the notochord, and the lateral mesoderm. We talk about somites that are an example of segmentation that is a pattern that is universal feature in animal development, and that we will see appear later during the semester when we talk the Hox gene and other type of segmentations examples. We also talk about kidney development as a model for organogenesis as a whole, and in this case of reciprocal induction. And finally, we talk about the importance of signals from neighboring germ cells and reciprocal interactions to help to form organs and structures. And we not only did that, we talk about how these produce different cell behaviors like the EMT and the MET. With this, we finish this section. And if you have any questions, just let me know. We can talk during class. Have a good day. Bye.